Hi, everyone. Thank you so much for joining us today virtually for the VTLSS seminar series. And I'm pleased to introduce Dr. Yusuke Sekine, an assistant professor from the University of Pittsburgh at the School of Medicine. So Dr. Sekine performed his graduate training at the University of Tokyo in Japan, studying how reactive oxygen species induce cell death mechanisms via the interference of particular phosphatase proteins that regulate apoptosis signaling, regulating kinase 1. And then he was awarded 2 postdoctoral fellowship awards to study at the University of Cambridge with Dr. David Rahn, where he studied the integrated stress response, in which this work was published in Science. And then he worked as a research fellow at the NIH in the Aging Institute with Dr. Torin Finkel to study the molecular biology of metabolic stress responses. And in 2018, he started his own laboratory. And I'm excited to hear about the work that's going on now that he's started his own independent group about the P53 transcriptional stress response and how membraneless organelles detect stress. So thank you so much for coming, Dr. Sekine, and we're excited to hear your talk.
Yes, thank you so much, Alicia, for your kind introduction, and thank you for this great opportunity. So I would like to talk today 2 topics, both related to our interests. That is how small molecules, such as pharmacological compounds and a physiological metabolite, impact cellular stress responses. First topic is about a chemical inhibitor called Isrid, Isrid, which inhibits the stress response called the integrated stress response. This study was mainly done in the David Rohn lab at the University of Cambridge when I was a postdoc. And the second topic is about today's seminar's title, the relation between acetyl-CoA metabolism and the stress response through the organelle nucleolus. Then the first topic, the integrated stress response. Here, I just called ISR, is the stress response to modulate mRNA translation in stress cells. In healthy cells, the translation initiation factor EIF2, which consists of 3 subunits, is catalyzed by its guanine nucleotide exchange factor EIF2B from a GDP bound to a GTP bound form. And only this GTP bound form of EIF2 can associate it with initiator methionine transfer RNA and 40S ribosomal subunit. And this tertiary complex can initiate translation. When cells are exposed to various stresses, some stress-activated kinases phosphorylate the serine 51 residue of EIF2 alpha. And this phosphorylation inhibits the catalytic cycles between EIF2 and EIF2B, thereby attenuating global translation. On the other hand, the translation of a small subset of mRNAs can be conversely promoted in this condition. This mRNAs includes the transcription factor ATF4, which is known as a master regulator of the ISR transcriptional program. So the ISR can be seen as a response to reallocate the translational demands from the general translation to the selective translation for stress adaptation. Because translation is very energy consuming process. This process can be also reallocation of energy consumption. So in mammalian cells, there are 4 EIF2 alpha kinases which are activated by specific stresses. For instance, the ER resident kinase PARC can be activated by the accumulation of unfolded protein in the ER. And PKR is activated by various RNA species. And GCN2 is activated by amino acid starvation or ribosomal storing. So like this, many other stresses can be sensed by either of these EIF2 alpha kinases. And these kinases phosphorylates are common substrate, EIF2 alpha, as I mentioned. Therefore, cell can induce a common translational control under various stress conditions. In 2013, Peter Walters' group found a chemical inhibitor of the ISR. It was named Isrid History. Here you can see the effect of Isrid in AR42J cells. Like an ER stressor tapshigarvin attenuates translation, which was monitored by the puromycin labeling, as shown here. And Isrid can reverse this translational attenuation as PARP inhibitor does. But unlike PARP inhibitors, Isrid does not suppress phosphorylation of ER2 alpha. This indicates the target of ISRIB Isrid is downstream of ER2 alpha phosphorylation, but upstream of translational attenuation. So it was quite likely that the Isrid targets the interaction between EIF2 and EIF2B. Either Isrid stimulates the catalytic interaction between EIF2 and EIF2B, or Isrid suppresses the inhibitory interaction between these two complexes. And indeed, we revealed that the Isrid can stimulate EIF2B gap activity using in vitro assays. However, at that time, we did not know the exact target and molecular action of Isrid. So to identify the target of Isrid, we took a cell genetic approach. We prepared a reporter cell line in which we can monitor the activation of ISR through the induction of GFP under the control of CHOP promoter. CHOP is the target gene of the ATF4. Here is the induction of the CHOP GFP reporter after treatment of the histidinol, which activates GCN2-mediated ISR. This induction is reversed by Isrid, as you can see here from orange line to the green line. So then to do a cell genetic screen, we mutagenize cells with chemical mutagens, such as EMS and ENU. And by using the flow cytometries, we screen cells that became resistant or insensitive to Isrid by these mutations. Repeating the selection enriched the Isrid-resistant cell population, and eventually we could isolate several clones that are partially or completely resistant to the Isrid. Here I showed two examples of Isrid-resistant clones. And then we isolated mRNAs from these cell lines and sequenced mRNAs for the EF2 and EF2B subunits to check whether they have any mutations. Actually, EF2B has 5 subunits, and we found mutations in one regulatory subunit, EF2B delta. And interestingly, these mutations are clustered at the N-terminal region of EF2B delta, as shown here. And so this suggests that EF2B delta, particularly this N-terminal region, may be a target of Isrid. But this random mutagenic approach could not exclude the possibility that other mutations that happened in the other genes also contributed to the Isrid resistance. So to exclude this possibility, we introduced each EF2B delta mutations into the parental reporter cells using CRISPR-Cas9 homology-directed repair technique. And this experiment demonstrated that mutations in EF2B delta, for example, L180F mutation, is sufficient to confer Isrid resistance to the cells. So 3 years later, David Long group and Peter Walter group both published a cryo-EM structure of human EF2B that is possessing Isrid inside the structure. And it shows that EF2B has a symmetric structure that consists of 2 sets of 5 subunits. And this structure clearly shows that Isrid binds to the center of heteromeric complex of 2 EF2B delta, shown in the green here, and 2 EF2B beta, shown in the light blue. And importantly, this Isrid binding site is at the center of entire EF2B structure. So this structure also shows that a human leucine 179, which corresponds to the hamster leucine 180, that I showed in the previous slide is indeed associated with Isrid in the structure. Also, further studies from several groups have established a current model of EF2 and EF2B interaction. So trimeric EF2 complex shown in grayscale here, and the decameric EF2B complex form two conformations. One is a catalytic conformation that promotes guanine nucleotide exchange cycle on EF2 by EF2B, and this promotes translation. And another is an inhibitory conformation with phosphorylated EF2 alpha. This conformation suppresses translation and induces ISR. So it appears that the binding of Isrid to the center of the EF2B antagonizes the phospho EF2 alpha induced inhibitory conformation, thereby promoting EF2B catalytic activity and that can inhibit ISR in the cell. Also, many groups have demonstrated that Isrid exhibits a diverse effect in vivo. For instance, the Isrid improved memory formation in rodents with various conditions. And also, Isrid prevents various neurological defects seen in several mouse models of neurodegenerative diseases. So suppressing ISR by Isrid seems to be beneficial for certain conditions. And so Isrid may be a good seed for developing therapeutics for neurological disorders. And I believe the original group who found this compound is doing this effort. Okay, so then I will move on the next topics about nuclear stress response. So currently our lab at the Pittsburgh is focusing on the mechanisms of how a mammalian cell senses and responses to the fluctuations in the intercellular metabolites. Here, I would just say metabolic stress for the changes in the metabolites, as metabolite fluctuations can be a kind of stress condition. A variety of factors induces metabolic stress, such as diet, fasting, exercise, and many cellular stresses, including hypoxia, and also biological rhythms and aging and various diseases. So metabolic stress activates signaling networks and stress responses to adapt to the stress conditions and maintain cellular and organismal homeostasis. Here, I want to emphasize that the number of studies have demonstrated that subcellular organelles such as the nucleus, endoplasmic reticulum, and Golgi lysosome and mitochondria play important roles for cellular metabolic stress responses. This is partly because organelles are highly associated with metabolic processes. Particularly, organelle membranes are important in the metabolic stress response, not just as a barriers for compartmentalization for metabolic reactions. Organelle membranes are known to serve as a platform for signaling to activate metabolic stress response. For instance, Brown and Goldstein lab has shown that the SCAP SREBP system exists on the ER membrane to sense cholesterol levels and maintain the cholesterol homeostasis. Also, it has been shown that the mTOR kinase can be activated on the lysosomal membrane in response to several amino acids changes, which promotes translation and cell growth. Also, recent studies, including our study, indicate that membraneless organelles, such as the nucleoli, as shown here, are also playing very important roles for sensing and responding to the metabolic stress conditions. Membraneless organelles are biomolecular condensates that are not surrounded by lipid bilayers. These condensates are formed through the process called liquid phase separation, LFPS, which I want to talk more detail later in this talk. So as these organelles do not have membranes, they are very sensitive to changes in their environment, including metabolic conditions. And they can very quickly respond to these changes. Today, I will focus on our recent study about the Nucleolus, which appears to be very important for cellular responses to changes in acetyl-CoA levels. Before going to the detail, I'd like to discuss some general principle of metabolite sensing and some difficulties to study metabolite-dependent cellular responses. So how metabolites can be sensed in cells? There are potentially two mechanisms. One is through the direct interaction between a metabolite and a protein, which can selectively bind to the metabolite. In fact, there are numbers of proteins or protein modules that can directly bind to metabolites, thereby acting as a metabolite sensor. These molecular sensors have a binding affinity to their partner metabolites within the range of physiological concentration of the metabolites so that they can react to physiological metabolite fluctuations. The interaction between sensors and metabolites alters their functional state of the functional state of the sensor proteins by changing proteins conformation, enzymatic activity, and protein interaction, localization or stabilization, and so on. And these changes become a signal to downstream effectors to induce cellular responses. The second way to sense metabolite is through metabolite-mediated post-translational modifications, such as saturation from acetyl-CoA and also methylation from methionine. And now we know that many other metabolites can be used for the post-translational modification in the cells. In this case, enzymes that catalyze the modification can be sensor proteins, but also many cases, metabolite-mediated post-translational modification can be processed without enzymes, therefore more directly reflects intracellular concentration of the metabolite. These modifications can again change the target protein structures and functions, thereby inducing the signal. So as you know, the post-translational modification of histones are extensively studied. However, those modifications on non-histone proteins have not been fully elucidated. So we are interested in the mechanism of how metabolic stress can be sensed and which signaling pathway is initially activated in the cells. However, it is not easy to study the metabolite-specific responses because, as shown here, the metabolic pathways are very complex and highly interconnected. So particularly when we want to identify specific responses to a particular metabolite or a particular metabolic pathway, we have to selectively manipulate the metabolite or the pathway, ideally without affecting other metabolic pathways, at least during the time scale of investigation. Also, metabolites are often compartmentalized by organelles, so it is also important to modulate the metabolite in a compartment-specific manner. So the problem to overcome is how we can selectively manipulate the metabolite, hopefully in a time and space controllable way, to understand the cellular responses to changes in a particular metabolite. For proteins, we have now many genetic tools to modulate the protein expression to understand the protein function, such as siRNA knockdown and CRISPR knockout, and many other overexpression systems. Also, however, I think experimental tools for metabolite manipulation is still limiting. Most commonly, we can use cell culture media that are deprived of specific nutrients such as glucose, lipid, or amino acid. They are useful, but these nutrients are metabolized to various metabolic intermediates and end products. So the effects are quite diverse. To regulate more specific metabolic pathway, we can use chemical or genetic inhibition of, for the specific biosynthesis pathway of the target metabolites, using gene knockout or knockdown of the enzyme that generates the metabolites, or chemical inhibitors for the enzyme, if such inhibitors is available. And recently, several groups have developed elegant methods to selectively control metabolites using bacterial enzymes. For instance, bacterial methionines that can specifically degrade methionine was used to induce methionine restriction in the flies. Also, other group utilized bacterial enzyme bacterial NADH oxidase for modulating NAD and NADH ratio. And also bacterial glutathione synthetase is used to increase glutathione levels. So importantly, expression of these enzymes can be controlled in time and compartment-specific manner by attaching signal sequence to the target organelles. These enzymes can be expressed in an organelle-specific manner. Also, by combining some inducible system, the timing of expression of these genes can be also regulated. So I think these are very good systems to selectively modulate target metabolites. The limitation is whether we can find a good enzyme in bacteria or other species for all of metabolites of interest. So lastly, one way to selectively manipulate metabolites is reprogramming of metabolic pathways. This can be seen as a kind of a synthetic biological approach and a bit artificial. And also it may be not easy to apply for every metabolic pathway. But using this approach, we made a cellular system to modulate cytosolic acetyl-CoA levels by extracellular acetate. I will move on the detail of this story. So, as you know, acetyl-CoA is an essential metabolite for the synthesis of various biological materials, including ATP and NADH in the mitochondria, and fatty acid and cholesterol in the cytosol. And acetyl-CoA is also utilized for the protein acetylation as a direct acetyl group donor. Acetylation regulates many protein functions and epigenetic regulations. And cellular nutrient availability directly influence acetyl-CoA levels. So it can be considered that the level of acetyl-CoA is an indicator of cellular metabolic states. Although it is well known that the acetyl-CoA levels are tightly linked to the histone acetylation levels, entire cellular responses to acetyl-CoA fluctuations are not fully understood. So in mammalian cells, acetyl-CoA is produced in two separated compartments. One is inside the mitochondria, and the other is outside of mitochondria, which includes cytosol and the nucleus. Mitochondrial acetyl-CoA is generated through the glycolysis or fatty acid beta oxidation or glutamine metabolism, and is used for the acetyl-CoA. It's used for NADH or ATP production, and also mitochondrial protein acetylation. But acetyl-CoA cannot pass the mitochondrial double membrane. So in the cytosol, acetyl-CoA is generated from citrate, which is exported from the mitochondrial transporters. Citrate is catalyzed by the enzyme called ACLY. Nucleoside-cytosolic acetyl-CoA is used for the lipid synthesis, such as fatty acid and steroids, and also for the protein acetylation. So in healthy cells, which are rich for the glucose, oxygen, and nutrient, and most of culture cell is quite healthy. And in this case, the citrate ACLY pathway is a main pathway to generate acetyl-CoA. But mammalian cell has another enzyme called ACSS2. ACSS2 makes acetyl-CoA using the metabolite acetate. In some cancer cells where the citrate-dependent acetyl-CoA production is impaired due to less nutrient or oxygen. So in that case, ACSS2 plays an essential role in acetyl-CoA production. and lipid synthesis and acetylation for the tumor development. Also, recent reports suggest that some type of cells, like immune cells and neurons, also use the acetate for acetyl-CoA production and histone acetylation for the specific gene expression. So altogether, the cytoplasmic and the nuclear acetyl-CoA levels are mainly maintained by these complementary pathways. So to analyze cellular responses to acetyl-CoA fluctuations, we first try to make an experimental system that we can selectively manipulate cytoplasmic acetyl-CoA levels. And to achieve it, we focused on this HLY and HSS2 complementary system. As the HLY pathway is essential for the culture cells, the knockout of HLY gene causes severe cell growth defects or death. However, we found that the supplementation of excess amount of acetate in the culture media could rescue the effect of HLY knockout and the cell grow normally. These are HLY knockout cell lines that are maintained in the high amount of acetate containing media, which we called acetate supplemented HLY knockout cells, acetyl-CoA cells. We also prepared HLY wild type control, which are cultured in the same 20 millimolar acetate containing media, but possessing the wild type HLY. Indeed, the survivability of this acetyl-CoA cell is depending on the acetate. If we remove acetate from the culture media, in other words, if we change the media to standard non-acetate media, we could observe a significant decrease in cell viability of acetyl-CoA cells, but not in the wild type cells. Because FPS contains several hundreds micromolar of acetate, when we use 1% FPS or 10% diaryzed FPS to remove the acetate completely, this effect was enhanced, as you can see here. So then we measured acetyl-CoA levels using the quantitative HPLC analysis and confirmed that the 4 hours acetate depletion drastically decreased the level of acetyl-CoA in cell lysate of acetyl-CoA cells. Conversely, the level of coenzyme A was increased. But these changes are not observed in the AC wild type cells. So we considered that these acetyl-CoA cells could be used as a good experimental system to analyze acute acetyl-CoA dependent cellular responses, because cytosolic acetyl-CoA is now regulated only through the acetate in the culture media. If we deplete acetate from the culture medium, cytosolic acetyl-CoA is also depleted. So importantly, we can address acute cellular responses within a few hours, which may be good to find an initial response to acetyl-CoA depletion, including the sensing mechanism. We combined this system to multiple omics analysis, including RNA-seq, proteome, and acetylome. And also we conducted CRISPR genetic screen to identify genes that are involved in this response. So we first checked the downstream event of acetyl-CoA metabolism. And during this acute, just 4 hours acetyl-CoA depletion, total lipid levels, including like cholesterol and various fatty acids, were not significantly changed. However, we found that the protein acetylation were quite rapidly decreased, as represented here by the histone acetylation and the alpha tubulin acetylation. Those are almost diminished within 30 minutes after acetate depletion. So we confirmed this using acetylome analysis, using a cell lysate after 90 minutes of acetate depletion. As shown here, the more than half of acetylation sites we detected appeared to be deacetylated, while a small subset of acetylation were increased in this condition. These potentially deacetylated proteins are involved in diverse cellular processes, including ribosomal RNA processing, translation, cell adhesion, mRNA splicing, and transcription or chromatin modification and so on. So it is likely that the alteration in the protein acetylation, mostly deacetylation, is a quite rapid response to acetyl-CoA depletion. We also conducted RNA-seq analysis and quantitative proteomics to detect global changes in mRNAs and proteins in response to the acetyl-CoA depletion. So in this plot, x-axis shows the changes of proteins which are increased or decreased upon acetyl-CoA depletion. And their mRNA changes are plotted along y-axis. By this plot, we found numbers of proteins that localize in the nucleolus, which are shown in red here. And these nuclear proteins are regulated at protein levels, but not mRNA levels. So we focused on the nucleolar changes upon acetyl-CoA depletion. As I mentioned at the beginning of this talk, the nucleolus is a membraneless organelle and known as a site for liposomal subunit biosynthesis. The structure of nucleolus is very unique and it consists of 3 layers, as you can see here. In the core component, FC, fibrillar center, is the place for the liposomal RNA synthesis. And the DFC is surrounding FC, shown in the yellow here. And it is a place for the liposomal RNA processing. And the most outside component, GC, granular component, is the site for the assembly of liposomal RNA and liposomal proteins. These layered structures are formed through the phase separation, and it shows liquid-like properties, which is important for the flux of liposomal RNA from FC to DFC to GC to proceed liposomal RNA maturation and assembly with liposomal proteins. So in the top panel, we marked the nucleolide with the UBF antibody, which is a transcription factor for the liposomal RNA. And also we stained liposomal RNA itself. And we found that upon acetyl-CoA depletion, the structure of nucleolus was drastically changed. Under steady state, ribosomal RNA and UBF are mostly co-localized, as you can see here. But upon acetyl-CoA depletion, total RNA level, ribosomal RNA levels were decreased, and the residual signal of ribosomal RNA appears to be segregated from UBF. So we also measured ribosomal RNA synthesis using the fluorouracil FURD incorporation assay. FURD incorporation into the ribosomal RNAs were detected by BRDU antibody. And we found that the acetate depletion significantly suppressed ribosomal RNA synthesis in the nucleolus. This observation suggests that the acetyl-CoA depletion alters nucleolar structures and suppress ribosomal RNA synthesis. In the same plot as the previous slide, we can also see the transcription factor P53 is induced by acetate depletion. We confirmed this P53 induction using immunoblotting. In assay wild type cells, there are no changes in the P53 levels. But in assay knockout KO cells, P53 was induced after 4 hours of acetate depletion with both 10% or 1% APS conditions. Also, RNA-seq analysis reveals that the several P53 target genes, including MDM2 or CDKN1A, were upregulated in acetate depleted cells, as shown here. So this suggests that acetyl-CoA depletion activates P53 dependent transcription. So now we observe the changes in the nucleolus and also P53 activation. Indeed, these two events are known to be linked quite closely. As I said, the nucleolus is the site for ribosomal biogenesis under normal conditions. However, a series of studies have indicated that the nucleolus is also playing a role as a stress-sensing organelle for activating p53-mediated stress responses. Various factors, including cellular stresses and also inhibition of ribosomal RNA synthesis by actinomycin D, or mutations in some ribosomal proteins, suppress the process of ribosomal biogenesis. This suppression triggers redistribution of unassembled ribosomal proteins and also some other nucleolar protein to the nucleoplasm. Most well-characterized example of this redistribution is 5S RNA RPL5 RPL11 complex. It is called 5S RNP complex, which is a part of the large ribosomal subunit under normal condition, but it is released into the nucleoplasm under stress condition. Then this complex directly binds to the MDM2. MDM2 is the E3 ligase for the p53 degradation at a steady state. So this interaction between the MDM2 and 5S RNP complex inhibits MDM2, thereby stabilizing p53 and activates the p53 target genes. This process is called the nucleolar stress response. In this process, the nucleolus serves as a signaling hub, converting various stress conditions to a response mediated by p53 activation. So this appears to be distinct from the mechanisms through the DNA damage induced p53 activation. But the common thing is MDM2 is anyway inhibited, either by phosphorylation by DNA damage activated kinase or by free ribosomal protein in the case of nucleolar stress response. So we consider that this acetyl-CoA depletion also induced the nucleolar stress response. Here we confirm that the acetate depletion induced the interaction between MDM2 and RPL11, as shown here. And the right panel shows that the knockdown of either RPL11 or RPL5 suppressed the acetate depletion dependent p53 induction. So these observations suggest that the acetyl-CoA depletion induces the ribosomal protein dependent p53 activation. Namely, acetyl-CoA depletion activates the nucleolar stress response. So next we asked what caused nucleolar stress in this context. Because as I mentioned earlier, the acetate depletion induced rapid deacetylation of many proteins. So we tested whether the deacetylation process is involved in the acetate depletion dependent p53 activation. We used various chemical inhibitors for histone deacetylases, and we found that a general inhibitor for the HDAC, such as trichostatin A or borinostat, completely suppressed the p53 induction upon acetate depletion, as you can see here. And we also used further selective inhibitors for each subclass of HDAC family, and this revealed that the selective inhibitors for the class A HDAC family, which includes HDAC 4, 5, 7, and 9, attenuated acetate depletion dependent p53 induction. So I show here that the class 2A HDAC inhibitor TMP195 suppresses the p53 induction upon acetate depletion. In contrast, the class 1 HDAC inhibitor entinostat did not suppress this induction. So also, class A HDAC inhibitors like TMP195 or NMK235 also restored the acetate depletion dependent suppression of FURD incorporation, that is ribosomal RNA synthesis, in the nucleolus. Also, but the class A1 inhibitor did not suppress, did not recover this suppression. So these observations indicate that class A HDAC mediated deacetylation causes nucleolar changes to trigger the nucleolar stress response. So to further understand the role of deacetylation by class A HDACs in the nucleolar stress response, we try to identify the substrate of class A HDACs. Indeed, their substrate have not been well characterized, but previous studies show that the class A HDACs are not good deacetylates for histones at least. So we conducted acetylome analysis using the class A HDAC inhibitor TMP195 and identified multiple candidates for substrate whose deacetylation were inhibited by TMP195. And so this list contains several nucleolar and liposomal proteins. And we first focus on the RPL11. Two lysine residue sites. So two lysine acetylation sites in the RPL11 were found to be TMP195 sensitive. So as RPL11 is crucial for P53 activation in the nucleolar stress response, we investigated the status of acetylation in RPL11. So immunoprecipitation of RPL11 using acetyl-lysine motif antibodies revealed that the RPL11 is indeed deacetylated upon acetate depletion, and this is reversed by the TMP195, but not by the class 1 inhibitor entinostat. So also, we mutated the lysine 67, which is close to the binding site of the RPL11 and MDM2. So lysine mutation to the glutamine, which mimics acetylation, attenuated acetate depletion induced interaction between MDM2 and RPL11, as you can see here. But it did not affect the interaction between RPL11 and RPL5. So this suggests that the acetylation of K67 is important to promote the interaction between MDM2 and RPL11, or the 5S RNP complex, which is central for P53 activation. We also focused on the NCL, which is also called nucleolin, and this is a scaffolding protein in the nucleolus. NCL has an intrinsically disordered region. It's called IDR at its N terminus and RNA binding motifs in the C terminus. And both domains are shown to be important for the maintaining phase-separated compartments. We found a clustered acetylation sites within the N-terminal IDR region, which were partly TMP195 sensitive. Because biological phase separation is caused by multivalent weak interactions between the various proteins and the nucleotides, as illustrated here. So post-translational modifications, including acetylation, can affect the entire status of the phase-separated compartment by influencing each weak interactions. So therefore, we tested whether the class A HDAC-mediated deacetylation affects the nucleolar phase separation. In other words, the liquidity of the nucleolus, which is important for the nucleolar functions. To address this point, we performed a FRAP analysis to monitor molecular dynamics within the nucleolus, which is an indicator of the liquidity. We measure the fluorescent recovery after photobleaching of NCL-GFP reporter within the nucleolus. This experiment showed that the acetate depletion accelerated the recovery time of NCL-GFP from the red to the green line. Maybe it's easier to see in the graph. Like the acetate depletion accelerated the recovery time. And this suggests that the molecular dynamics of the NCL is enhanced in the nucleolus after acetyl-CoA depletion. This suggests that the class A HDAC-mediated deacetylation, and then The class A HDAC inhibitors, especially LMK235, can reverse this acceleration, as you can see here. So this suggests that class A HDAC mediated deacetylation enhance the mobility or liquidity of the nucleolus, which might promote the distribution of ribosomal protein to the nucleoplasm. Although we need further confirmation. So this is a summary slide for this study and also my talk. We propose a model as the following. So in high acetyl-CoA status, nucleolar proteins are acetylated to some extent, which is important for the steady state function of the nucleolus, including the ribosomal RNA synthesis. When acetyl-CoA levels goes down, class A HDACs deacetylate some nucleolar and ribosomal proteins, which modulates the nuclear phase separation, as well as the interaction between MDM2 and RPL11. These changes suppress the ribosomal RNA synthesis and also promotes p53 mediated stress responses. So this model indicates that the nucleolus, membraneless organelle, plays a crucial role in sensing metabolic stress through metabolite mediated post-translational modification. So now we are trying to extend our findings to more general principle. Not only the nucleolus, various membraneless organelles or biomolecular condensates can be regulated by metabolite mediated post-translational modification, which play important roles in the metabolic stress responses. So the last slide is an acknowledgement. Actually, we are still a small group. Shiori is a co-PI of this group, and Ryan is our research technician. And I'd like to thank especially the director of Aging Institute, Torren Finkel, for many help of our project. And also, I also thank many collaborators in Pittsburgh University, as well as also in the NIH. And I'd like to thank Lone Lab members in the University of Cambridge for the previous work. Thank you for your listening.
Yusuke, thank you so much. If you have a question, please put it in the Q&A box and I will read it to him. I actually have a question about your work. One thing that I didn't see is recovery. So when you add back acetate to these cells, can you recover this p53 response? Do we see that the cells can recover? And is there a point where the cells can't recover any longer?
Yeah, I haven't done the detailed experiment, but it's sure that if you can put back acetate in the cells, you can rescue the cell death at least. But I haven't seen the signal, the p53. Like we can, because once p53 is activated, yeah, it should be downregulated if we put back acetate. But I don't have that data. Yeah, that's very interesting point. So I also want to try like which metabolite can rescue the phenotype, like not only acetate, but maybe many other like metabolites that can produce acetyl-CoA should be able to rescue the phenotype. So that's very interesting point.
Yeah. So one other question I did have too is about stress granules. Does this happen also with stress granules if you added like oxidative stress to these particular cells? Would you see that these stress granules would act differently and not really coalesce and make the what's it called, like with the liquid phase separation?
Yeah, so that is a very interesting point. Actually, I tested several phase separated components like stress granules and also nuclear spectrals. Actually, I think stress granules were a little bit increased upon acetate depletion. So I want to definitely check the more detail about the stress granules. And also, some nuclear spectrals are also changed upon the acetate depletion. So I think definitely, yeah, various phase separated compartments are changed during the acetate depletion. So that's, yeah, what I want to study
Okay.
Yeah.
All right, so I'm looking to see if there's any other questions for your talk, but thank you so much for sharing your new work and your new story with us. Yeah, I don't see any other questions, so we'll go ahead and sign off. If you have a question, you can always email me and I can forward it to Yusuke at a later date. And you have your individual meetings this afternoon. So thank you so much for joining us for